Good afternoon, everyone. I'm very happy to present our work on predicting anatomical tumor growth in pediatric high-grade glioma patients, and we did this using denoising diffusion models. So let's get straight into it. There are around 80 cases per year in Switzerland of pediatric patients suffering from central nervous system tumors, and around half of these are gliomas. Among these gliomas, there is a particular subset, quite a small subset, that has a very aggressive phenotype and also quite a poor prognosis. And despite the fact that these are quite rare, they are responsible for 40% of the brain tumor-related deaths in children. And this kind of patients usually go through multiple rounds of magnetic resonance imaging, which is the standard in this case, and this is done for diagnosis, but also as follow-ups during their treatment and to inform actually treatment. Most of the time, this treatment is radiotherapy, since this is the only one available given the characteristics of this type of tumors. And when we think about this, the radiotherapy design is also based on identifying regions in the brain where the tumor is likely to expand. And keeping this in mind, if we would be able to predict this anatomical tumor growth regions, then we could also think of a personalized treatment and which could positively impact the outcomes. Another point, and very important one, since this is such a rare disease, you can imagine in the context of a clinical trial, you would have to have a clinical, the treatment branch and also a control branch. It's anyways hard enough to get enough patients that are to take part in such clinical trials. So having accurate predictions would enable us to use such predictions as synthetic controls. So we would use these predictions as our control branch in the clinical trial. And with this, we set ourselves to explore how we can use computer vision models, particularly guided diffusion models, to generate such predictions of anatomical tumor growth in the form of MRI scans. So how did we do this? We used guided diffusion models. For those who attended earlier the AI in genomics track, you probably got a bit of an introduction to this. But what they do is they learn a denoising distribution of the data. And you can see here in the bottom part, this is the denoising part. What we did, apart from just learning the distribution of the data, is to change some of the feature in the input image. So right here, we wanted to change the tumor size, and we did this by introducing this guiding term that enables us to actually generate a tumor MRI scan with a tumor that is bigger than the ones we started from. To develop the model, we used MRI data from the BRATS challenge, so the Brain Tumor Segmentation Challenge, and we used actually both adult and pediatric patients. This is important since we are talking about a rare disease. We don't have enough data to train such deep models just using pediatric data. So what we did actually is we performed transfer learning. You've just heard about it before. And in this case, it worked because it was imaging data. So we first trained on the adult, and then we performed transfer learning to pediatric patients. What I just want to stress here is that we had one time point. So we actually just had an input scan, and we made it grow from the available time point. And then once this was working, we wanted to validate our results, and for this we used quite a small data set of pediatric patients where we actually had longitudinal data available. So we had the MRI scans nicely showing the progression. Well, the progression of the tumor is not nice, but you could really see how the tumor would grow, and we could compare that to what our model predicted. And in this case, actually also one important part is that we only had one MRI contrast available, so just the T2 flare. Now I want to show you the results of how this works. You can see here examples of both adult and pediatric patients and how the tumor grows. If you're having trouble to really identify the parts where the tumor grows, you can have a look at the difference map. And there are a few things to point out here. The first one is that the tumor expands around the edges of the initial tumor, which is what you would expect to see in reality. And also, you don't see any kind of abnormal growth pattern. So in none of the samples that we inspected visually, you could see the tumor growing into the ventricles, which is something that you also don't see in reality. So we were happy to see that the model learns a realistic growth of the tumor. And since we were expecting to validate this only using one of the MRI contrasts, we also looked at a very at an equivalent model. In the first case that you've seen, it was trained using all possible MRI contrasts that we had available. And for this part, we only used the T2 flare. Visually, they're quite similar. You can probably see, for example, that the tumor growth is not as localized and as well defined as when the model gets more information from the other contrasts. Apart from generating an MRI scan with an enlarged tumor, the very nice aspect of these models is that they have a probabilistic nature. So when you start from slightly different initializations, you can obtain slightly different results. And to have some more robust predictions, we actually repeated this generation multiple times, and we managed to create this tumor growth probability heat maps where you can interpret each pixel, the color of each pixel, as being the probability of that pixel turning from a non-tumor pixel into a tumor pixel. And I just want to say that, yeah, obviously upon visual inspection, everything seems to be working fine, but we also need to quantify this. And in this case, we used the two very common image quality metrics, the FID that looks at the distance between the distribution of the real data and of the generated data. And then we also looked at the structural similarity between real and generated images. You can see here the results. For the FID, we're looking for the smallest value possible, and state-of-the-art generative models usually score between 10 and 15 in the medical imaging context. But I just want to mention that this is highly sensitive to the data set size, which is why you see a bit of variability. However, for the structural similarity, we see high results in all models, and both for adult and pediatric patients, which was encouraging. Apart from this, we also actually had some trainee radiologists, some medical students, to look at generated and real images and try to tell them apart. And it was, fortunately for us, very hard for them to do so. So they only managed to do this with a 51% accuracy, which is basically random. So this was also a very good sign that the images we generate are realistic. And on top of this, we investigated radiomic features as well. These radiomic features are based on on the value of the pixels. There are some simple ones, such as, for example, the mean pixel value, and they go all the way to more complex ones, such as the uniformity or the neighborhood gray level tone and other more complex features of the images themselves. And we observed that only 10% were significant between real and generated, so 90% of them were non-significant between the two sets. And then the final test, we produced these enlarged tumors in the MRI scan, but how do they compare to the ground truth? What you see here on the bottom with blue is the evolution in tumor size of a patient where we have this longitudinal MRI scan, so you can see how the tumor is growing, and you can also see what our model predicted and how it compares to it. So if we look at week 25, and at the error map as well, we see that it manages very nicely to recapitulate kind of the tumor growth. When we go a bit further, it doesn't perform as well, but however, we can see that the growth along the side of the ventricle is kind of identified, and there is a lot of potential to look into. We see here that the tumor sizes match, so just really the area, but what we also looked at was the dye score, which actually quantifies the overlap between the segmentations of those tumors, which was around 0.8, so that's 80% of the pixels were correctly identified by our model to be exactly the ones that we saw in the ground truth. And as I've shown earlier, we also computed these tumor growth probability maps that, apart from some artifacts, again, quite nicely recapitulate the tumor growth. And with this, I just want to say that this was a proof of concept study, but we were very happy to see promising results and the potential of using such models to predict anatomical tumor growth. There are obviously many other things we want to look into. Right now, you need to specify how big the tumor you want to be. You want it to be in the final image, also to extend validation, and many others. And with this, I just want to thank everyone in the lab at ETH, and you may recognize some familiar faces today, but also to all of the other people who contributed to this work, and special thanks to the DMG Center Zurich, Leonhard Med, and Olga Mayen Fish Foundation. And thank you very much.